Hour 19. Welcome, Mitchell.
Thank you, Sandra. Bonjour. Good morning.
Bonjour. So for everyone out there who doesn't know, this is Mitchell Warren. He heads, he's the fearless leader of AVAC, Global Advocacy for HIV Prevention. And I work with Mitchell at AVAC. So Mitchell, I'm going to hand it over to you. You're going to do your thing, and I'm going to sit back and watch you.
Fantastic. Sandra, thank you. And thank you for Hour 18. That was brilliant. Your own work with the transgender scorecard, hearing Yvette describe work in Asia, and the remarkable Linda-Gail talking vaccines. And the through line, I think, is from Glenda, an icon in South Africa and the world. We're now moving on to another South African researcher as part of our next panel. This is Hour 19. We are now 2/3 of the way through this marathon. And what Brian Manalga said just in his remarkable testimonial, it's about science, it's about truth. And it's been a remarkable journey for 44 years. It's been a remarkable journey for 18 hours. I am so excited about this Hour 19 because we're going to, in the next hour, talk about another very specific journey. It's the journey from basic science into product development and clinical research, and hopefully the next chapter soon to be written, how we translate great science into public health impact. And we're going to do this all in a conversation about something you've heard a lot about these past 18 hours, and that's injectable Lenacapavir, the newest PrEP option. And I want to bring in 3 people who are just a remarkable trio who represent a much larger community who've really pioneered work over the last 2 decades or more, which you'll hear about. So I'm delighted to welcome Wes Sundquist from the University of Utah, Linda-Gail Bekker from the Desmond Tutu Health Foundation in South Africa, and Yvette Raphael, another fierce and fabulous South African. Wes, this is 2 American guys and 2 remarkable South African women in conversation. Yvette leads Advocacy for Prevention of HIV and AIDS in Africa, APHA. And what brings the 3 of you together in this platform is the story of Lenacapavir. And we're not going to so much focus on the details of every clinical trial that was done, but really talk about how we got to whatever, September 2025 on the precipice of something, but how it began many, many years before, actually, Wes, with some of your research with colleagues funded by the NIH. And we want to start with you, Wes, and then we'll go through with Linda-Gail and Yvette in a bit of a trajectory of where we go next. But let me turn to you, Wes. And I know you are, lucky for you, you are not home in Utah, where it would be, I guess, 3 a.m., but I know you're dialing in from Prague at a conference, and we're so glad you could make time to join us.
So thanks, Mitchell. You can hear me fine?
Perfectly.
Great. So let me start by saying thanks to Mitchell and to AVAC for organizing this really important event. I'm Wes Sundquist. I'm a basic scientist who studies HIV at the University of Utah in the US. And I guess my job is to set the stage for this session by talking a bit about the science behind this new drug, Lenacapavir, which was developed by Gilead Sciences. So it's a pleasure to share this session with Linda-Gail and Yvette, who are 2 people I greatly admire. And we have very different expertise, so this should be fun. Lenacapavir is a very potent and long-lasting antiviral therapeutic and PrEP drug, and it targets the viral capsid. So we're about to talk about the capsid. I need to share my slides. Do I do that just by clicking on them? Okay. So these are electron micrographic images of HIV particles that show what the virus looks like at near atomic level. And the virus has 2 states. So it first comes out of cells in what we call the immature state. That's the state shown at the left. And that's the viral machine that's capable of exiting cells, but it's not yet infectious. So under normal circumstances, the virus then undergoes a process that we call maturation, which creates the particle on the right that can now infect a new cell. And that transformation from left to right requires the viral protease, and the protease inhibitors actually block this step, which is how they work clinically. So as you've undoubtedly already noticed, the most striking feature of the mature infectious virus is the formation of this central cone-shaped container that we call the capsid. And if you can load my video, but not quite start it yet. Oh, there we go. That's perfect. So this is a video that shows how the HIV capsid is created during viral assembly and maturation. And it was made by my very talented colleague, Janet Owasa, who's a molecular animator. And so at the bottom of the slide is an infected cell, and the space above that is the area into which the virus is going to leave the cell. And so lower is the cell, upper is the extracellular space. And if you start the video, what you can see is that these orange molecules, which are the structural protein of the virus, capture the viral RNA genome. So that's shown in blue. The virus has to leave with its genome, and you can see it making a particle that's leaving the cell. And as you get to the stage where it's still tethered by a membrane to the cell, the virus recruits host factors that now pinch off the neck and release the viral particle. And that's the immature particle that gets released. So in a moment, you'll see a pink molecule, which is the protease. It cuts itself out and then also cuts the structural proteins. And those proteins then rearrange quite dramatically during maturation to create the viral capsid that surrounds the RNA genome. And so it's this yellow object, the capsid, that's the target of Lenacapavir. So that's what we're going to be talking about. And we now understand this process of virus assembly in great detail. And that's really due to hard work on the part of many different research groups around the world. As Mitch said, I'm coming to you from the Retrovirus Assembly meeting, which is in Prague this year. And we're gathered here to debate all the molecular details of the process shown in Janet's beautiful video. Scientists are funny. We care passionately about every detail. But if we sort of step back, then the take-home message is really that just how much we know. Thank you. I can go to my slides. Just how much we know about the structure of the capsid and the process of HIV assembly in just over 40 years since it was all discovered. And that information was generated almost exclusively by publicly funded basic research. And the information has really been critical for identifying steps in the viral replication step that are vulnerable to new inhibitors. So let me make sure I can advance. Yep. So on the left, you see a panel that shows a molecular model for the viral capsid. And we now understand how the viral proteins come together to make the cone-shaped capsid. And we understand the positions of the thousands of atoms that come together to make the cone. In the center, on the top, you can see the structure of the drug Lenacapavir. And it binds in a pocket that's repeated across the surface of the capsid. And that pocket is shown in the box in orange. And an important component of that pocket is that it's relatively invariant. That means that strains from all different continents have the same pocket, and that means that Lenacapavir is active against many different viral strains. If we go to the next slide, what you see here is a schematic of the HIV replication cycle. And so the movie that I showed you focuses on the right side where the virus was being made. But once the virus is made as an infectious particle, it then has to infect a new T cell. And so if we look at the top, we see an infectious viral particle. It's going to recognize a new T cell and go on to infect it through interactions with the receptors, which are called CD4 and the co-receptor. And that starts the first half of the viral life cycle. And I don't want to go into that in detail, except to say that 4 important things have to happen. One is that the capsid has to enter the cell. The RNA genome, which is shown in light blue, has to be copied by the viral reverse transcriptase to make a double-stranded DNA. That's shown in purple. The core particle, the conical capsid, has to make it into the nucleus, and the DNA has to integrate into the host chromosome. And the fact that the virus integrates its DNA into the host chromosome is the reason that it's so hard to cure HIV from infected people, because every time a cell gets infected, it archives a copy of the virus. And so what I want to talk about now is work early on. We and others made mutations in the protein that makes up the capsid. And what we discovered was that even small changes in the capsid blocked different steps of the viral life cycle, and those are shown in the T-shaped schematics throughout the viral life cycle. And so the surprise was that the virus really cares about the capsid and that the capsid functions at many different steps of the viral life cycle. So if we now look at Lenacapavir, we see that it blocks many of those same steps that are capsid dependent. So it's most potent at blocking the process in which the capsid gets into the nucleus. That's shown in the lower left. But it also blocks the processes of assembly and maturation and even reverse transcription at high concentrations. And so part of the magic of Lenacapavir is that it blocks multiple steps in HIV replication. I should pause for just a moment and say that the other drugs that we have in the clinic work on the first half of the viral life cycle. So they block reverse transcription of RNA into DNA and also the integration of that DNA into the host chromosome. And again, they were developed by public-private partnerships, much the kind that I'm talking about for Lenacapavir. Okay, so I want to finish with my last slide, emphasizing the really important role that Gilead played in developing Lenacapavir. So this is a slide that I borrowed from Thomas Silar, who's Senior Vice President for Virology Research at Gilead. And in this slide, he starts by emphasizing the importance of some of the foundational publicly funded studies that I've been describing, which convinced him and ultimately convinced Gilead to start mount a very large capsid inhibitor program. And I think that was really quite visionary on his part and on Gilead's part. So as you can see that from the timeline, they started that program in 2006. And they first did what's called high throughput screening. So that is designed to identify chemicals that are in their inventory that would bind weakly to the capsid and inhibit its functions. And that was under the direction of Steve Yant. And they identified inhibitors and worked on them for 3 years, but ultimately decided that they weren't going to be successful. So they started over with a second generation set of compounds. And at that point, those are the molecules that eventually led to Lenacapavir. And I want to emphasize what a heroic effort that was to make their weak inhibitors stronger. So that was led by John Link, and they made more than 4,000 compounds. And in each case, they tested their not only their efficacy, but also their stability and their pharmacology and their off-targets effects. And another magic of Lenacapavir is its pharmacology. It lasts for a very long time in the body, and that's how it can last so long after a single injection. That coupled with its solubility. So you can see the trend in the green dots. Each of those is a compound that they made, and they go lower as you go to the right through time. And that's because lower are more potent compounds. And so they made compounds that were ultimately about 100,000 times more potent than their original molecules. So I just want to end by emphasizing a few key points that might not be obvious to people who aren't familiar with drug development. The first is what I've said earlier, which is research that was done in the public sector really was critical for convincing Gilead to initiate their capsid inhibitor program. The second point is that it took guts and also vision to tackle the capsid as a drug target, because capsids are actually a non-traditional target for reasons we can discuss in the Q&A. And so there was skepticism it would be a good drug target, but Thomas Silar and Gilead believed in it. The next thing is that Lenacapavir itself is a very non-traditional drug. It's much larger and less soluble than most drugs, and that turns out to be very important for its long-lasting efficacy. And so we can talk about that in the question and answer session. And then finally, the commitment that Gilead had. So you can see that they funded this project for more than a decade before they got to a clinical candidate. And so all of this was quite a heroic effort, and in my opinion, it wouldn't have been possible to do in an academic lab because we generally lack the resources and also the expertise to do this kind of work. So it really did take a public research and private partnership to make Lenacapavir. So when they got to this point, they still needed to test the candidate drug in the clinic and in the community. That took another 9 years to get to the present, and that's where Linda-Gail and Yvette take over the story.
Wes, I have to say, when we put this whole concept together of 24 hours of AIDS research. Our goal was to have science made approachable to the general public, and your 13 minutes and the Academy Award for, I didn't even know there was such a term as molecular animation, but I learned more in that video than I think I've known for my whole career. And thank you for making that possible. And we'll pick up on a number of things. 1996, was that kind of the first publication from you and your colleagues that really catapulted us forward?
Yeah, maybe even 1994. So it's striking that that's 30 years ago. And one of the things we need to do is to learn to do this faster, but it emphasizes how important it was to have long-term support through that entire process. Otherwise, we and others couldn't have done what we did.
Absolutely. And we'll come back to that in the conversation. It is so striking to see that we're in these decades. It just may be coincidental, maybe not. 96 to 06, from kind of real basic science before the 06 to 2016 period of developing the molecule. And maybe that's the perfect place, Linda-Gail, because I believe it was 2016, 17, 18 PrEP research, which you pioneered in so many different domains, was already delivering oral PrEP, and the vaginal ring results came out in 2016. Cabotegravir was in the clinic in 2016 as well. But if you want to step in now and give us a sense of when you first heard of Lenacapavir, and maybe take us on that next decade of the journey.
Thanks, Mitchell. And yes, just to say it's a pleasure to be in hour 19. It's been extraordinary to get snippets of the first 18 hours. So thank you to all the colleagues who've just stepped forward. I'm so proud to be part of this research ecosystem and the contribution that is made to optimizing global health generally. And thanks to Wes and Yvette for being part of this panel. It's such a pleasure always. And Wes, yes, you get the Emmy Award, I think, for the making science palatable. And again, congrats for this beautiful molecule. And yes, so we take the banner. We're kind of the middle section. We take the banner, the next step on before we pass off into scale up and roll out of a product that will hopefully change lives and change the epidemic. We believe this is a game changer. So I became first aware of this notion of injectable PrEP as a concept, which I work and I have worked for 30 years with young people who face the real risk of HIV. In my region today, 570 young women acquired HIV or will acquire HIV, 570 in a single day. And that really has been our story right from the get-go. Of course, across the world, there are another 570, 580 young people in all kinds of walks of life that will acquire HIV. So there is no doubt that primary prevention is absolutely key to bringing this epidemic under control. The first 30 years were all about treatment, and rightly so. We needed to save lives and bring death under control. We now need to end this epidemic. And the only way we can do this is with primary prevention. You're right, Mitchell. We've had oral PrEP. It works. It's brilliant. We need to roll it out. We've got it to about 10 million people around the world, but it is not enough. It is not enough because people struggle to do something daily in their lives. And enter Lenacapavir, an injectable monthly PrEP agent, which we believe is going to be a game changer. Why? Because this is where clinical research is really important. So initially, there always is preclinical work to make sure that this is safe to move into humans. We then move into humans through phase 1, phase 2, making sure it's safe for humans to tolerate. And then we have to ask the question, does this effectively stop HIV, prevent HIV acquisition? We also need to be sure that it is safe when given to a wide range of humanity. And this is where the purpose programs designed by Gilead in consultation and in partnership with the community, which Yvette will beautifully expound on, but also in partnership with clinical researchers around the world. Now, there are at the moment in the suite of studies of purpose, 5 studies making sure we really are testing this product in a very wide range of people, everybody, across the world who may find themselves in a position where they are at risk of acquiring HIV and who need this product are being exposed to it, so we can understand how well it works for a variety of people, including people who may be pregnant, people who may be lactating, and so on and so forth. Now, 2 registrational trials were conducted, Purpose 1 and Purpose 2. These are important because these are the pivotal trials on which the regulators can decide whether this is something that can be rolled out across the world, and that leads to approval. And that needs very good, believable, credible, trusted data in order to make those decisions. And that means you've got to go to sites that can do world-class, internationally recognized, international standard research in order to make that decision. And indeed, Purpose 1 was the study that was conducted in adolescent girls and young women who have sex with men. This was only possible to do in a region such as mine. I am here in Cape Town, South Africa. Purpose 1 was conducted in South Africa and Uganda in 28 sites, in sites that had been developed, the infrastructure, the know-how, the capacity was developed through 35, 40 years of solid research to understand how antiretrovirals work, how we can use them more optimally, how TB interacts with HIV. When the COVID epidemic came along, we pivoted to show that we could use COVID vaccines. And indeed, it is that infrastructure that provided the foundation to say, will Lenacapavir prevent HIV in young women should they be exposed to an HIV virus? So that was Purpose 1. Purpose 2 was similarly conducted in sites all around the world in a variety of populations, cisgender men, transgender women, non-binary people who have, again, at risk in all kinds of walks of life. That was Purpose 2. These 2 trials asked the simple question, will Lenacapavir protect people against HIV? And indeed, the results have been legendary. And I must say again, this is where the molecule needs to be celebrated, Wes, because it truly has delivered in that regard. So Purpose 1 showing that in the woman who received Lenacapavir, we did not see a single infection during the randomized controlled trial phase. In Purpose 2, there were just 2 infections. So really amounting to a more than 98% efficacy for both, for really every population around the world. But I want to say, Mitchell, for the adolescent girls and young women, it was profound, because this population, we have really struggled to find a prevention modality that works, all the time for everybody. And it has been extraordinary to be able to share those results, not a single infection in the randomized trial phase. And so that amounted to 100% efficacy. It was compared to background HIV incidence through a well-designed study. And of course, it showed superiority to oral PrEP, not because oral PrEP doesn't work. It does work, but it has to be taken consistently. And what we found in the trial, as we have found in the real world, is that young people often, with all of the issues they have to deal with every day, struggle to remember to take a pill on a daily basis. And so the wonderful story is that Lenacapavir is now ready and poised to be rolled out in the real world to as many people who can use it. in order to bring this epidemic under control as a public health threat. And we are very excited about the prospects of moving now from the phase 3s, the pivotal trials, with the evidence that they work, the approvals from our regulators, to then move into the general public, assured by good science, strong, robust clinical research, that this product is safe and efficacious and will do its magic. And so with that, I think it is time now to hand off to Yvette with the understanding that none of this can happen unless we have strong partnerships with communities, that we do this in sync and with the trust and belief of communities. And this, again, is the magic of research. It truly is a multi-sectoral, multi-party activity, and absolutely critical to advancing science and optimizing healthcare. And so over to you, Yvette.
LGB, brilliant. And, just as you said at the end, science moves with the speed of trust. We learned that in COVID. We're seeing it play out in real time in a negative way when we lose that trust and confidence. But Yvette, who's a dear, dear friend and colleague, leads not only APHA, but also established with a number of leading African women advocates, the African Women's Prevention Community Accountability Board, APHA, is just a pioneer in this. And Yvette, I remember thinking about this journey. It's not as if the researchers turn to the community now in 2025 and say, we discovered something, what do you want to do? I remember being with you in 2018 and 2019, so a number of years ago, and APHA in the African AIDS Conference in Kigali, Rwanda in 2019. Would love to have you pick it up when you first encountered this concept of Lenacapavir and what it means and what it should mean going forward and what we do about it all.
Am I unmuted?
You are unmuted and clear and wonderful.
Thank you so much. Good morning, good afternoon, good evening, everyone. Thank you again for inviting me to be part of this very important and much needed global action to save science. I have titled my talk, How Women Sold Science to Our Community. I also want to acknowledge my mentors who sparked the idea that HIV prevention and research is possible. I bring into this space the many women who've sacrificed so much for research to be successful, and also the many young people whom we, as their mothers, have promised a future free of HIV. And then we promised them 2030. I know the upper ground forces are watching, so hello guys. I want to start my talk at the beginning. My own diagnosis 25 years ago, a woman with a small baby. I just started my new job. I started feeling very tired and lost a lot of weight. Getting tested and starting ARVs at the height of denialism in South Africa, taking ARVs at a time that it was so demonized. I would like for the backstage people to please play my video. It's important that this story, a 50-year-old woman says she's living with HIV, stops with me, that this will not be the story of our daughters. My daughter turning 25 was not only a trigger, but it was a reminder of my own journey, that I was exactly her age when I was diagnosed with HIV. Just after I gave birth to her was I diagnosed with HIV. I was very worried that she might follow in the same path. I actually used my own HIV status to do a dig at HIV. I'm a beautiful Black woman. I'm happy, I'm healthy. I take my meds sometimes. I struggle to take my meds, and I tell that story. I just try to be as authentic as I can. I fought for HIV awareness in the workplace. So my whole journey about HIV is just about learning, teaching people living with HIV, but also fighting sometimes. I'm always on the sidelines fighting for the rights of people living with HIV. My life was saved by treatment. I was one of the very few first women in South Africa who took HIV treatment in 2000. So I am passionate about science. I'm passionate about what science can do for us and for women. So when we got an email from the Gilead team that they want to have an urgent meeting to tell us the results, we were so excited. We had to ask this question over and over. What does the 100% mean? And it meant that no one in the trial was infected with HIV. And we wanted to know, when is it going to become available? And we have been speaking about speed with Gilead during the Accountability Board term. And this would mean our girls, young people, women in Africa will have access within the next 2 years after the news. And now our daughters have an opportunity to not have the same story that we have. A story of, hi, my name is Yvette and I'm living with HIV. Our daughters can just be themselves. Engaged in sub-Saharan Africa prevention research was only the participants and their community leaders. So the GCAGs really were aware and are aware that women living with HIV, women impacted by HIV work in solidarity with the community-based advisory boards and with each other, with recognition of specific relevance of our views and their views and expertise. Every trial study now has an advisory board, and that they engage outside of their own cabs and sites. So for the choice manifesto, for each trial we worked on, we would advocate for the method to be vigorously available, and then funders would begin to say that the next product would be better, especially when that product was less dependent on the user adherence. We saw people say that oral PrEP didn't work in women, when what they should have said is that oral PrEP works in women who take it regularly. We saw the funders tell us that the ring wasn't worth having in the toolbox. We heard the description of trial participants who could not adhere, that made those participants seem unreliable or non-compliant, instead of being women sharing information about what worked for them right at that moment. So with the Choice Manifesto, it is also a reflection of a research-engaged feminist community that believes in support and champions research, but does not support decisions about what women want being made by anyone but the women. The Global Community Advisory Accountability Board assured that the voices of women are heard from the trenches. We remembered Shakira for asking with such authority why women are not given the right to choose if they want to be part of studies, whether pregnant or breastfeeding. We also remembered how young women had to wait to start PrEP and the use of the ring because they were not part of the initial studies. These delays in access did not help in bringing down the incidence of young women, which was our clarion call. The PEPFAR cuts are hurting research badly. So much recruitment is from the communities, clinics, or start with HIV testing. Some people embedded in clinics supporting the work of identifying potential partners who lost their job. Testing is collapsing in so many places. So when we talk about resourcing clinical trials, we also need to talk about resourcing the healthcare with HIV prevention and treatment in the surrounding communities, my community. Thank you.
Yvette, I hope you can see at the bottom one of the many young women who you are mentoring and leading. And I had the privilege of being with you last week, and it's everyone talks about Auntie Yvette, and you just have shown the world why that is. And I'm going to pause there before we go into a conversation, because this moment is too important to let pass. I should say that I think the video was remarkable, and I don't think I'll ever see it too often, Yvette. And it came from the award ceremony from Science Magazine, which had recognized Lenacapavir as the breakthrough of the year in 2024. But this year, you and Wes and the Gilead team under Mopli Das, who led the clinical development on behalf of Gilead, and the research teams around the world, including Linda-Gail, were presented with the award for it being the breakthrough. And I think what you three have demonstrated is why that is. It's not just about the science. It's about, as you said, when communities lead. There was a session many hours ago, I forget which hour it was, where a comrade said, community did this. And that's true in every part of our 44-year odyssey, all of us. But Yvette, what you just captured is community did this. Couldn't do it without Wes and basic science, couldn't do it without Gilead Sciences, couldn't do it without Linda-Gail and clinical trialists around the world. But community, and not just advocates, but community. And I think, Linda-Gail, you said as you started that you're happy to be part of this community. And Glenda said that too last hour. Community did this. Yes, it was Desmond, Dixon, Diallo, Danielle. It is staggering to see and hear. I do want, Yvette, you said something toward the end about what the devastation of these cuts mean, and I want to look at this in a number of different ways. Wes and Linda-Gail, there have lots of things are on the table or on the chopping block right now. One is basic science, and one is international research and research in South Africa. And I know you both talked about it, but I wonder, and then I'm going to come to you, Yvette, about what the chopping block means to deliver on the promise of science. But Wes and Linda-Gail, 30 years ago, Wes, you were doing the basic science, Linda-Gail running clinical trials for decades. What's the future look like to you if some of the draconian cuts being proposed of basic science and global research happen? Well, what does it mean? Maybe, Wes, you want to start?
Sure. So I think this is a worrisome time for us. I'm a department chair, and we have a lot of talented young faculty members, and of course they're trainees, and they're all wondering what the future looks like. So maybe I'll just talk a little bit about our own journey. So as you said, we started this 30 years ago, and all of our work was consistently funded through those 30 years by the National Institutes of Health in the US. And I would emphasize that that's quite a competitive process. So throughout that 30 years, every time we applied for a grant, we had to convince people that we had made progress, and about 1 in 5, sometimes 1 in 10 grants are funded. So it's not as though it's automatic. But nevertheless, we had the sense that if we did good work, that we were going to be funded and could continue the work. And that's just critical for tackling long-term projects like understanding the HIV capsid. And so I think in some sense we were lucky the capsid didn't have to end up being a good drug target. But I think if you look across basic research, what you find is every time there's a breakthrough, someone was lucky. But if you integrate across the entire field, you say that basic research consistently delivers new ways of thinking about therapeutic modalities and new ways of thinking about human health. And so I think individually we can't, we don't know what we're going to find, but what we can promise is that as a field, we will deliver new ways of thinking about things. And I guess it's, without going too far in the weeds, the Senate has passed a bill that looks quite promising, but the House is much less so, and the administration's proposals are even more concerning. And this really would have prevented young researchers like I once was from tackling big, hard problems. And then, of course, it's important once those breakthroughs are made to translate them, and that's where Linda-Gail's done such amazing work.
And Linda-Gail, before you jump in, I want to remind everyone on this daveaidsresearch.org website, there is a section to take action. What Wes just said, NIH has consistently had bipartisan support, as has PEPFAR. Take action. If you're inspired by what you've heard from Wes and Linda-Gail and Yvette, if you're American, call your member of Congress. If you're not an American and you're in a country with an embassy, tell your embassy that NIH matters, that research matters. Those voices matter. So make those calls, write those letters, be in touch with your members of Congress to say, thank you for supporting NIH. Thank you for supporting PEPFAR, because they need to have the winds behind them to have the courage to stand up when the administration has said no. But Linda-Gail, the clinical side of things, you've had decades of support too. What does the future look like?
Thanks, Mitchell. I think I want to start by saying, the international community does this in partnership. It's very important to recognize that this is not a one-way street. This is a bi-directional street. And of course, the dollars, the federal dollars, make all the difference, and the scale of the amount of money that we need to be able to really do this internationally recognized research is huge, and sadly often beyond what our own countries can deliver, although we continue, as I heard Glenda saying in the last hour, we have to keep encouraging our domestic governments to step up and contribute to research in general, and in clinical research in communicable diseases more specifically. But I do want to emphasize that we have, particularly with our collaborators in the US, with the NIH colleagues, we've done this in partnership. This has been co-designed research where we have put our minds together to say, how do we advance the science in such a way that we globally make a difference to this epidemic? It is, after all, a pandemic. It is affecting every single country in the world. And therefore, it makes sense that we have joint efforts to solve the problems. And so over these many decades, this infrastructure has been built, as I say, in order to meet the goals of both the global north and the global south. And that has been extraordinary. And it is a jewel that now we cannot afford to lose. Because, not only can we take this infrastructure and very quickly, I want to say, if it took 20 years to get Wes's molecule to the point where it could go into humans, it's taken a very short period of time to now get to the point where we have FDA approval for this product. And that's because the wheels are oiled. We are an awesome set of machinery ready to go. And we showed this during the COVID-19 pandemic. When the COVID-19 pandemic hit, we pivoted in a dime to say, here's a new threat to this planet. What can we do jointly to move this forward? And it was extraordinary what we could achieve. And here we are again today saying, we want to continue to do this work in partnership because it is the right thing to do. It is the most efficient way to do it, and it is likely to have the best success. And so when you ask me, what does it look like if we don't fix this? I would say a huge dearth of good science and good research coming. I see a void. I see difficulty getting the kinds of success we've seen in the last 20, 30 years, that not happening. And that is something that should keep us all up every single night, worrying about our collective futures.
Linda-Gail, thank you for that. We have to remind people, acronyms are in our field everywhere, but we have to remind people that the NIH is the National Institutes of Health, not the National Health Institute. It's not the U.S. Health Institute, it's the National Institutes of Health. But even for those who are very U.S.-centric, is it safe to say, and you were involved, you're the only, I think, country, South Africa, that was involved in both Purpose 1 and Purpose 2. The FDA, the US regulator, approved this product within a year of the first Purpose 1 result, is it safe to say that without South African research sites, we'd still be sitting with Purpose 1 and 2 for years to come without answering the question?
Totally, Mitchell. 25 of the 28 sites were in South Africa. The other 3 were in Uganda. The contribution from these 2 countries to the world cannot be undervalued. We have done it in record time to international standards. We have brought the result. Thank you also, again, multiple partners in getting this achieved, but it could not have been done without South Africa. And that also goes to, good information about how to treat children with HIV, good information on how to treat TB and HIV as a joint problem, good information on how to optimize antiretrovirals, and so on and so forth. So again, a partnership, but a partnership that has made a huge contribution to the global knowledge and performance or response in this epidemic.
Yeah, no, American women will benefit from Lenacapavir because of African women leading. And so, Yvette, you got that call back in June of 2024, a year ago. The most beautiful number in HIV research, 0, no infections. What was your first thought? And what did you think would happen next, going back a year in time? And then where are we now?
Thank you so much, Mitchell, for that. And I think I remember just thinking no young person will be infected with HIV after this. And next was, are we going to have to wait that long again? Are we going to have to wait 5-6 years for it to be accessible, not only in the world, but South Africa specifically, where it will take a little bit longer. But then advocates started working with organizations like AVAC and many other organizations and thinking around how can we speed this up? We have enough information to not make the same mistakes. And we wanted a good story for HIV prevention. So what happened in January 27, 28 is nothing that we anticipated. And it was not the news that we needed to ensure that Lenacapavir becomes available in our communities for young people to ensure that they have a different future from what we had. So it was almost the same kind of shock going over my, what do you mean 100%? Nobody was infected, and that was what we understood, and that's what I understood, and that meant a future free of HIV for many, many South African girls. So I also want to just bring in how that impacted the community and some of the sites that we thought would be dependent, we would depend on to ensure Lenacapavir becomes available and accessible to South Africa. So many of those sites are at the brim, at the verge of closing down. Many of the healthcare workers are looking, all that information, all that expertise, are looking for other ways to provide for their family. I went to an organization yesterday and met a young boy who is living with HIV, but he's like, I don't understand, why am I taking medication and nobody else is taking medication? So the science is needed to ensure no babies are born with HIV so that they don't have the same struggles as that 14-year-old. So it's important for us to know that we have to fight to ensure this doesn't end. Everything depends on it. Everything.
That beautifully said. That none of this science happens to have a publication. It happens to impact people's lives. But Wes, back in 1994, you were doing the science with many colleagues. You were being published. Does a basic scientist think like, I'm doing this in the laboratory? Did you have a vision that there'd be a product at the end of this when you were first discovering all of this? I mean, what were you thinking in 94?
So day to day, most of what we do is figuring out why an experiment didn't work. So we're troubleshooting, but I do think it's fair to say that, when I started our lab at the University of Utah, we decided to work on HIV because it was a huge global. We didn't appreciate just how global a problem, but it was clearly an epidemic already in men in the US, and we now know it's a global epidemic. And so I think we did, and as did many other labs, I should emphasize the basic research community, just like the other communities we've heard about, is a lot of important people. We did realize that, what we did could have an impact in the longer run. So I think on a daily basis, we're trying to figure out how to solve problems, but worked on this problem because we thought if we and other labs like us tackle these problems, then we'll know things that could ultimately be therapeutically useful. And that's what's turned out to be true.
Fantastic. In terms of, you said, in terms of solving problems, and
just because we're running. I missed that, but maybe I'll make a point that I think that something that's really important for people to understand is there's still very important research to be done. So you've heard about the implementation science, and this is really going to be learning how to do things well. There can be drug resistance. What happens when that emerges? Is it a problem? What are we going to do? And then also at the basic level, there are really important questions. So we still can't cure people who are HIV positive, but we're making real progress, not our lab, but other labs. And so there's reason to think that, this can be something that can fall. And the same is true for vaccines, either prophylactic vaccines or therapeutic vaccines. So Lenacapavir is amazing, but it lasts 6 months. But for other infectious diseases, we can take a vaccine as a kids and are protected for our whole lives, or we can boost our immune system if we're already HIV positive. So I think people should not think that there is not a great need for research in the HIV field, because there is, all the way from implementation science to very basic questions in immunology.
Wes, thank you for that, because one of the things that I wanted to ask all of you, but maybe start with you, Linda-Gail. We talk, there's a real need to rebalance the portfolio at any moment. You have new insights, you might change the research you do. We've heard some in the NIH and in other settings say Lenacapavir is a miracle drug. And I'm a big believer that there's no such thing as a miracle drug, but some have said that. And there's some who would say, we need to stop. We don't need to fund basic science anymore. We don't need to fund vaccine research. We just should fund implementation science of Lenacapavir. And I wonder, I don't know who's watching out there. I hope leadership at the NIH is watching. If they are, what's the message, Linda-Gail? Lenacapavir is remarkable. Should we shift this pendulum from, should we say to Glenda an hour ago, sorry, Glenda, we don't need you anymore. We don't need a vaccine. We're going to do implementation science instead. What's the message that people need to hear?
Mitchell, a resounding absolutely not. We cannot stop this train. And the reason is, we saw clips of what the virus does, how it acts, and Wes, also alluded to this. This virus is constantly on the move, and therefore we cannot sit back and rest. Yes, we have a magnificent molecule that lasts for a very long time in the body. We can deliver it with an injection. There are other things in the pipeline, I'm glad to say, and Yvette has also alluded to the fact that when we offer people choice of prevention, they're far more likely to use what works for them, use it more effectively, we're more likely to have coverage. But there is no doubt, I'm with you, that this virus will find a way to get around a single molecule. And unfortunately that we've seen that, we all remember, well, I'm old enough to remember how excited we were when zidovudine was discovered and how that changed the face of an absolutely devastating life sentence where death was inevitable if somebody acquired HIV. But we learned very quickly that single action, single drug zidovudine had a very limited lifetime, and that's why we have to come with triple therapy. Similarly, we're going to have to have multiple prevention options, and as you know, I'm a great believer in vaccines. I'm an infectious diseases doctor. You cannot overcome an infectious disease without a preventive vaccine. And of course, the promise of a cure remains so important for the 40 million people around the world who live with this virus today.
Beautiful. Thank you for that, Linda-Gail. And I want Alice Seto, who's been a great colleague in helping to organize this whole marathon, in the chat quotes Paul Offit. I'd call it a miracle, except that it is explained by science. Science matters. And Yvette, you talked about, science and we talk a lot in advocacy about citizen sciences. Neither you nor I are trained as scientists, but citizen science is what has got HIV research for 44 years driving. I wonder if you could just spend a minute talking, one of the great innovations in the PURPOSE trials, both 1 and 2, was the inclusion of young people early. And one of Linda-Gail's colleagues, Catherine Gill, spoke a couple of hours ago about this. But there were the inclusion of adolescents and the inclusion and allowance for pregnant and breastfeeding women to continue in PURPOSE 1 in the trial. These were novel designs, which I don't think get talked about enough. They get kind of overlooked in the big headline news. Do you want to say a word not only about the design, but what your and colleagues' impact was in the good participatory practice aspect of this research?
Yes, thank you, Mitchell. And I think I touched a little bit about it, on why it was so important for us to make sure that we don't use the old methods to ensure that access happens speedily. So our engagement with Gilead was to ensure, when we did the, go through the documents, is to ensure that young women literally were part of this. And not only were young women part of the trials, but we also had to engage with the people who are their, guardians and make decisions of them. And Linda-Gail's team at DTHF with Nombeko and many other women held lots of sessions to ensure that the parents understand why the young people want to be part of the trial, but also what their kids are taking. So it is not just one engagement and making sure that we get people to go, but also how do we support them to ensure that they continue to be part of the trial, and then they stay in the trial so that the results that is needed come through. And also with the pregnant women and breastfeeding people, it was important that we, every time women have to be part of something, there's a hurdle. So what about the baby? What about the fetus? What about your husband? And all of those did not depend on a decision a woman would make. So we actually encouraged the trial to say, even though you're not going to say we're recruiting pregnant women or breastfeeding women, you would reconsent, basically going back to the woman and ask her for her agency to say, I want to continue because I want other women to benefit from this action today. And also how that was supported with engagement in the sites. Gilead gave us an opportunity to engage around the sites where we were, and also in making sure that these women know exactly what they do, what they are reconsenting to. So it was important. Actually, all trials are now using the accountability model, but also are thinking around including pregnant and breastfeeding women and young women. But it's just not something that is done for the sake of being done. There is a lot of investment that goes through that. And me and Linda-Gail had a discussion last week at the AIDS conference on how important that investment is, how important that engagement is to ensure the trial just doesn't tick boxes. And this was one of the trials that we didn't allow Gilead to tick boxes.
Beautiful. Yvette, thank you. And Desmond talked about this early yesterday. So at some point yesterday, I lose track of time. But about community did this, about the good participatory practice. I will put in a plug in the 9 o'clock Eastern time hour. So in about 2 hours, one of my colleagues, Stacey Hanna, is going to talk about good participatory practice and the lessons, particularly from the PURPOSE program. We are almost out of time. This conversation could go on for another 24 hours, but I want to give you each a sentence or two, a chance to just, what's the message to the community watching this that you want to leave people with? And let me go and I was doing the same order we started with, but Wes, why don't we start with you, live from Prague?
Sure. I guess, so two things. One is there's this saying, bench to bedside to community. And I think that was really captured in the presentations, that drugs get developed through a pipeline, and they're only as, it only works as well as the weakest link in that pipeline. So they all need to be supported and done exceptionally well. Maybe one other point that I think hasn't come out in terms of basic research is the sort of spin-offs. So, you know, we may never get to an HIV cure, although we obviously hope we will, but we're going to learn an enormous amount along the way. And Yvette just made the nice point that what we learned about how to do clinical trials in communities with young women and lactating women and so forth has knock-on effects for other health and clinical trials. And so this is true for basic science as well. What we learn in trying to develop an HIV vaccine transfers to COVID and will transfer to other infectious diseases. And so it's, I guess maybe the take-home message is that it's such an ecosystem, that science is an ecosystem, and so is the pipeline to get to improving human health.
Beautiful. LGB?
Mitchell, I'm going to be quick, and I'm going to actually invoke a very good, wonderful colleague who we lost, unfortunately, a decade ago, Steve Lawn, who did amazing work here, born in England, but worked here on TB and HIV. And he said the following. He said, it is not research, it is research. And we search for a way to improve global well-being. But I do want to say it's an international, domestic partnership, and we are stronger together.
Beautiful. Ecosystem partnership. Comrade, Yvette, last word.
Thank you so much. I wrote down a scientist, a researcher, and a village girl. It means that we all can come together, work together, and ensure that there is a successful outcome. It cannot be the end of research, and we will continue to fight. I always say this, in a revolution, you either go to prison or you die, and we are not going to die.
Hour 19, a basic scientist, a clinician, and a village girl walked into a bar. And while this is not a miracle drug, they made magic. And I am so grateful to all three of you and all the people you represent in each of your domains who got us here and who passed the baton to make sure that we don't leave this in a journal, but rather we make it come alive in people's lives. Thank you all three for all that you've done. And thank you all for staying through hour 19.